Okay, so exam number 1 is in the books now, and hopefully you learned some things from exam number 1. If you didn't do as well as you had hoped or expected, I hope you will take advantage of the bonus points. This is one of the few times we get bonus points in the class, so please make sure that you do that. A little bit of a reality check in that the first exam was a lot of review, a lot of that stuff you had covered last semester, and so now we're getting into a little bit newer material. So typically the second exam is a little bit more challenging than the first exam was, so that's something you just need to keep in mind. And big take home is start studying early. Okay. For example, there were a couple of questions that I put together from the study guide for that essay question on the endomembrane system. And some people really didn't understand what the endomembrane system was and confused it with phospholipid bilayer cell membrane structure. Okay, very different thing. If you had done the study guide questions, and particularly if you'd been checked with a mentor or with me, we could have caught that particular, misunderstanding, and that would have saved you some points. And so start working on the study guide questions. I like to give you some stats for the exam to review statistical analysis and to make sure that you understand, because you've got lab material to turn in, that you're properly interpreting statistics correctly. So we've got 3 sets of information up here, and here is your data by lab section, giving you mean, standard deviation, and the n value, number of people in the lab. Here are the t-value comparisons, and then here is an abbreviated t-table giving you certain degrees of freedom for 95, and that should be 7%. It should be 1%. That's 99% confidence. When you look at data and start trying to interpret things, mean is a good place to start looking. Okay, and so if you look at the mean, there seems to be a pattern, and there was a very good performance in the Monday lab section, for example. What's standard deviation a reflection of? Standard deviation is a reflection of variation of data. Okay, so how much spread was there within the people within that particular lab section? And you can see that the variation is almost twice as great in the Thursday lab section as the Monday lab section. Why is that going to be important? Well, that's going to be important down here. Here's our calculated t-values. So if I compare the mean of the Monday lab section to the Wednesday lab section and take into account the variation within the data, here is our calculated t-value based upon that particular comparison of Monday to Wednesday, 3.85. What does that mean? Well, when you start looking at this particular calculated data, which is derived from the numbers, the performance in the class, then this value has to exceed the tabular or expected t-value for the appropriate degrees of freedom. Okay? So is there a significant difference, for example, between the performance of the Wednesday lab section and the Thursday lab section? Well, if you just look at the means, you go, yeah, there is, right? But is that difference statistically significant? Okay, so how do we determine that? Well, we have to look at this calculated t-value. So the calculated t-value for Thursday versus Wednesday is 1.456. Okay, is that difference significant? That value would have to be greater than the tabular t-value for the appropriate degrees of freedom. What would be the degrees of freedom for this particular comparison right here? Wednesday lab versus Thursday lab. 12, right? So we look down here at 12. And if we want to be at least 95% confidence, this value would have to exceed 2.179. It doesn't, okay? So even though there's a numerical difference between these particular means, it's not a statistically significant difference. So make sure you understand that when you're doing lab data, for example. If we're comparing class mean values for pH 3 sucrose versus pH 5 sucrose, it's tempting to just say the one that had the highest mean is the one that ferments the fastest at that particular pH. But you can only make that conclusion if the difference in the means is statistically significant, at least at the 95% confidence level. If it's not, then even though those values are different numerically, we would have to say from a statistical significance standpoint that they tied with one another and that there was no real difference between the two. Does that make sense? Okay. Okay, for reading quiz on Monday, we will do reading quiz on just the last part of the chapter 6, which is enzymes. So sections 6.4 and 6.5, 125 through 132. And this will be a nice complement because you will have a pre-lab quiz, right?
Yes. The next lab on enzymes is a 2-week lab. And so the lab write-up in the manual has week 1, week 2. You will be responsible for the material of week 1, and there will be a quiz.
Okay. So there's going to be some parallel between the information for the reading quiz and the pre-lab quiz. You get 2 for 1. Okay, so we are in the process right now of talking about this somewhat confounding concepts of cells and entropy and the second law of thermodynamics. Where we left off last time was talking about, okay, what is a cell? Well, a cell is a bunch of molecules, proteins, carbohydrates, lipids, nucleic acids, that are organized in such a fashion that the molecules make up cell organelles, for example, and a cell is a bunch of cell organelles basically interacting with one another, giving us various processes that we associate with life. Okay? So cells are highly organized units. Remember, because entropy is a measure of disorganization, cells are states of low entropy. Okay? Well, what does the second law say? The second law of thermodynamics says that the natural tendency of matter is to move from states of low entropy, organization, to high entropy, disorganization. So the second law of thermodynamics basically says things get messy on their own. Okay? Well, if cells lose their organization, they can no longer carry out the processes that we call life, and therefore, by definition, those cells are dying or dead. Okay, so a loss of organization and increase in entropy is cellular death. Well, we can fight this tendency of getting messy or losing your organization by expending energy. And cells constantly having to expend energy to maintain their organization. It's constant work to stay alive. Do cells violate the second law of thermodynamics when they're doing this? No, because what the second law of thermodynamics does is it distinguishes between two things. Systems and surroundings. Or, if you like, cell/organism and environment. The second law of thermodynamics says that overall entropy increases. Does that allow for little pockets of low entropy like a cell or an organism? Yes, it does, as long as the entropy of the overall environment correspondingly increases. So we can have low states of entropy if the environment basically pays for that. Okay? So overall, the entropy of the universe is constantly increasing. We've got an expanding universe, for example. Okay? But that does allow for pockets of low entropy, life, but those living organisms are existing at the expense of increasing the disorder of their surroundings, making their surroundings more messy. Okay? So life isn't contradicting the second law of thermodynamics. Cells take in highly ordered molecules, food, and use that to provide the energy to maintain their organization, their low state of entropy. But in doing so, they generate waste products which they dump off to their environment. Okay? The reason that cells can do this is because they are a type of system called an open system. A system is considered to be open if it can exchange energy and matter with its environment. If a cell were a closed system, closed systems can exchange energy and matter with their environment, then things fall apart. So the only reason that life can exist is because there's this ability, because of the open nature of a cell, it brings things in, it dumps things out. That allows it to maintain its low state of entropy, basically at the expense of its surrounding environment. Can a closed system maintain a low state of entropy? No, because closed systems doesn't have the ability for this interaction. They can't bring energy in. They can't get rid of waste products. So here's a good example of both laws of thermodynamics in a kind of common occurrence. We've got a cheetah and its prey down here. And as this cheetah is feeding on this organism's tissues, it is consuming the organic molecules, proteins, carbohydrates, lipids, nucleic acids, that make up the cells of that organism. And it is converting the chemical energy of those molecules into ATP, which it's then using to maintain the organization of the cheetah cells. Okay? We're not creating energy, we're not destroying energy, but we are changing it from one form of another. In this case, one form of chemical energy, glucose, for example, into another form of chemical energy, ATP. Okay? What does the second law of thermodynamics tell us? It tells us that in order for this cheetah to stay alive, for it to have organized cells, it's going to have to expend energy. ATP energy. Okay? What happens when it expends that energy? It causes the entropy of its environment to increase. Why? Because the environment provided this chemical energy to begin with. Highly ordered molecules. And what does the cheetah cells do when they burn this chemical energy like sugar? They release less ordered, simpler molecules like carbon dioxide and water. So they took from the environment complex molecules and they made less complex molecules as a waste product. Some of that ATP energy didn't get converted into meaningful work. And the cells produce a fair amount of heat energy as a waste product. What do organisms have to do? They've got to shed that heat. Otherwise they would get too hot, cells would die. And so it has to release heat from the environment. So the highly ordered chemical energy was taken from the environment, and what was returned to the environment? Less ordered, simpler molecules like carbon dioxide and water, and waste forms of energy like heat. So this cheetah is living at the expense of its environment. It's an open system. It can exchange energy and matter with its surroundings. So everybody's happy. No laws of thermodynamics have been violated. So what does the second law say about life? Well, basically it says that life is improbable. Because what's the second law of thermodynamics say? Normally, everything gets messy. But life's not messy. Life's organized. So life's kind of improbable. Life cannot violate the principle that entropy increases. That's one of those laws of science. But living things are able to live at the expense of their environment. They maintain their state of decreased entropy because they're increasing the entropy of their surroundings. So overall, if you take into account the environment and the organism, overall, the entropy of the total universe is still increasing constantly. But life can only do this if it's constantly expending energy. And there's a special form of energy that a cell typically uses to do that, to maintain its organization. What's the form of energy? It's kind of the energy currency of the cell. ATP, right? So we need to look at ATP a little bit and see what it looks like, what it's made up of, and then talk a little bit about something called the ATP cycle, because that's going to be important. Okay, so here's ATP. ATP stands for adenosine triphosphate. Here is the adenosine portion, and here are the 3 phosphate groups. This look familiar at all? Where have you seen something that looks somewhat like this? Maybe this? What are these nucleotides that are held together? What are the 4 nucleotides? Adenine, guanine, thymine, and cytosine. And what does a nucleotide consist of? A sugar, a phosphate, a single phosphate, and a nitrogen base. Okay? So this energy molecule is very chemically related to the same thing, a nucleotide that stores genetic information. It's kind of interesting. What's the major difference? The fact that ATP has 2 additional phosphate groups attached to it. One of the things we typically equate ATP with is being a quote-unquote high-energy molecule. And again, sometimes people have the concept that, well, if it's a high-energy molecule, it's something like gasoline, for example. And if you burn it, it will release energy to do work, like it does in a car. A little bit different in cellular metabolism. What does the ATP cycle refer to? It refers to the fact that adenosine triphosphate, ATP, can be converted into adenosine diphosphate and an inorganic phosphate. This molecule can be hydrolyzed. It requires water molecules for the hydrolysis process to occur. When this happens, there is a release of energy. Most of this energy that's released is in the form of heat. Is heat a good form of energy for the cell to try to produce to do work? Cell wants to accomplish something. Should it just really make itself really hot? What's the problem with that? What happens when a cell gets too hot? Things start to denature, in part because hydrogen bonds are disrupted by increased temperature. And so DNA molecules start to fall apart. Proteins start to lose their 3-dimensional structure. Cells and organisms attempt to maintain a fairly constant temperature or an isothermal state. In our bodies, we have some pretty elaborate mechanisms to ensure we don't heat up too much. What are they? Sweating, for example. Okay? So where you're able to thermoregulate. It's really important for that to happen. So in cellular metabolism, heat is oftentimes a waste product. And if it's generated, it really can't be used to do any cellular work. And even more importantly, the organism or the cell's got to get rid of that waste heat or the cells will die. So a lot of the energy that was originally stored in this molecule is released as this waste product heat. And this is a much poorer energy molecule than the original was. Okay? But what we've seen is this reaction is important. Why? Because it liberates this phosphate group. And what can that phosphate group do? It can phosphorylate proteins. And that phosphorylation, changing the shape of those proteins, can accomplish cellular work like transport, or dynein walking, or contraction. Okay? Well, pretty quickly we would burn up all of the cell's ATP, so we've got to be able to regenerate that ATP to keep things going. How do you do that? Well, you can stick a phosphate back onto ADP and make ATP. But what is that going to require? An input of energy. Okay? So ATP is constantly in the cell being broken down or hydrolyzed and then being reformed again. When it's broken down, work can be accomplished. Its reformation requires an input of energy for that to happen. Now, this is going to get a little bit confusing, potentially. But let's talk about this for a second. Let's go back to this slide for a second. ATP hydrolysis, this process right here, is an energy-releasing phenomenon. What has to happen in order for you to go from here to here? Doesn't a bond have to be broken? Doesn't this bond have to be broken right here? So, bond breaking releases energy, right? Chemistry folks, is that correct? Does bond breaking release energy? Breaking bonds requires energy. What releases energy? Bond formation releases energy. So we seem to have kind of an inconsistency here, because I'm telling you on one hand, ATP hydrolysis involves bond breaking. But I'm also telling you that this process releases energy. Isn't that inconsistent? It is. But that's only part of the story. In reality, ATP hydrolysis involves both of these phenomena. Yes, some bonds are being broken. But as we'll see in a second, some other new bonds are being formed. More energy is released as new bonds are formed than is used up in breaking bonds. So if you look at this process overall, it's energy liberating or releasing, because more energy is released by new bond formation than is used up in bond breaking. So that's what this particular slide is all about. Take a look at it for a second. Here's ATP. And I just abbreviated the adenosine portion as an A, and then here's our 3 phosphates. This is a hydrolysis reaction. So it's going to involve water. Here is ADP, and here is phosphate. So, what's happening over here on this side? Some bonds have to be broken. This bond right here, this OP bond, has to be broken. That's going to require some energy. This bond between H and OH has to be broken. That's going to require some energy. Okay? Does it require a lot of energy to break these HOH bonds? No. It's a pretty fragile bond. Is it going to require a lot of energy to break this bond? As it turns out, no. That bond is very unstable. It's very fragile as well. That's why sometimes you'll see that bond represented not by a straight line, but a little squiggly line. And you'll oftentimes be told that represents a high energy bond. What that really represents is a very unstable bond, one that's easily broken. Okay, when we make ADP and phosphate, we form some new bonds. We form an OH bond, and we form a POH bond. Those bond formations release energy. The energy required to break the bonds is called activation energy. This energy that's released is referred to as enthalpy. Don't worry about those particular terms. But here's what you should understand. The amount of energy that's released is greater than that which is consumed in the process. By this amount, 7.3 kilocalories per mole. That's how much energy is released when ATP is hydrolyzed. So if we look at that particular energy little balloon right here, that represents 7.3 kilocalories per mole for ATP hydrolysis. That's how much energy is liberated by this particular process. It's primarily due to the fact that this bond is very unstable and doesn't require much energy to break. Okay? So we've already talked about this, ATP and cellular work. How does ATP hydrolysis power cellular work? Primarily by liberating phosphate, which can cause phosphorylation and subsequent dephosphorylation of proteins. So when ATP is hydrolyzed, the phosphate groups can cause motor proteins to function or membrane proteins to function. Phosphorylation of chemical compounds in metabolism is also important for making these particular reactions occur. Here's a chemical reaction, for example. Here is a compound called glutamic acid. It can react with ammonia to produce a new product called glutamine. What does that actually involve? This is an energy requiring process. It happens at the expense of ATP hydrolysis because the glutamic acid becomes temporarily phosphorylated. And that makes it more reactive to react with ammonia to produce the glutamine. So all this cellular work requires ATP, but most of the time, what's driving the work is the phosphate that's released that can then phosphorylate something. If we need to do more work, we've got to regenerate that ATP. We've got to stick these things back together, and that requires an input of energy. So, how do we make ATP? Well, the first way we'll talk about making ATP is by digging into the process of cellular respiration. And let's use as our example the aerobic form of cellular respiration. What are the reactants in aerobic cellular respiration? What's being burned up? What's the fuel for cellular respiration? A little bit of a trick question, because actually there are several potential fuels. But most of them involve converting one molecule, like a protein or a lipid, into glucose molecules. So when we talk about cellular respiration, we're usually talking about breaking down glucose. What's the other major reactant involved in aerobic cellular respiration? What does aerobic refer to? Oxygen, okay? And what's produced as we break down that glucose? We're going to produce some energy. And the energy can be and is ATP. What's another form of energy produced? Is all the energy of glucose converted into ATP? No. We generate a fair amount of heat. What are the other products that are produced besides forms of energy? What are the waste products? Caleb says carbon dioxide. That's correct. And what's the other one? And water. Sometimes this water that's produced is called metabolic water. There are actually some organisms that live in very arid environments that have to drink very little. How come? Because they have very elaborate water conservation methods, so they can retain their water really well. But they need water still. Where do they get it? From their metabolism? Okay. Where do organisms get their sugar to burn? Where do you get your sugar to burn? From your food that you eat, right? You might eat a real starchy meal. What's starch? It's a carbohydrate, and it's a monomer or polymer. It is a polymer of what? Glucose, right? And actually it is the alpha forms of glucose that are joined together to form starch. And we can hydrolyze those. We can digest the starch and form glucose, and then use the glucose in cellular respiration to make ATP. If we equate money with energy, ATP would be considered to be kind of the cash of the cell's energy systems. Cash is something that, at least for right now, you can still spend anywhere, right? Everybody takes cash. What does glucose represent? It's a different form of energy or a different form of money. It might be a check. Does everybody take checks? No, especially if you don't have the proper identification, right? Can cells use glucose directly to do cellular work? No. This can't phosphorylate things. And so in some respects, cell respiration can be kind of considered to be analogous to the check cashing process. You can go to a bank and cash the check and get money, and then you can spend that money to buy what you want to buy, or in the case of the cell, to do the work you need to do. What does starch represent in this particular case? Starch represents maybe a savings account. It's a stored form of glucose. You can't just go to a store and plop down your savings passbook and say, I'm paying for my stuff. They don't accept that form of currency. So the cell has to be able to convert chemical energy from one form to another, but ultimately it's all about getting this molecule which you can use to do the cellular work that's necessary. Organisms that get their energy for cellular respiration from other organisms are called consumers. We're consumers. This is a little bit of an oversimplification, but it's good enough for right now. Another term that is used to describe a consumer is a heterotroph. Troph means to feed. Hetero means others. These are organisms that get their chemical energy by consuming other organisms. Can everybody be a consumer and we have a functioning planet? What do we also need? We need some producers. What's another word for a producer? Some kind of a troph. It's an autotroph. These are organisms that feed themselves. They create their own glucose. How do they do that? They take carbon dioxide from the environment. They take water from the environment. And they take some ATP that they have produced using sunlight energy. What's this process of turning sunlight energy into chemical energy called? That's photosynthesis, right? And then they can stick these things together and make the sugar. Okay? So you see the dependence, the interdependence between autotrophs and heterotrophs, or consumers and producers. Okay? What organelle does photosynthesis happen in? Chloroplast, right? What organelle does aerobic cellular respiration happen in? Mitochondria. So, plants have chloroplasts, animals have mitochondria, right? Well, yes, they do, but do plants only have chloroplasts and no mitochondria? No. So plants not only have to be able to make sugar by photosynthesis, but their cells also have to have the ability to convert glucose back into ATP to do cellular work. What about animal cells? They need mitochondria, right? But they don't have chloroplasts. They're unable to be the producers. So when we go through cellular respiration first, we're going to go through it in a fair amount of detail. And if you master some of the concepts associated with cellular respiration, particularly understand how the ATP is produced by chemiosmosis. If you understand it for respiration, understanding it for photosynthesis is going to be pretty much a piece of cake. Okay? So those processes we'll talk about, those metabolic processes, are very interrelated to one another. Okay. A cell is basically a bunch of chemical interactions. The chemistry faculty would love to hear me say this, but it's really true. Life is chemistry. The chemistry is complex, and it's self-regulated. And we're going to see in a while that the regulatory aspect of metabolism is the result of the action of enzymes. Cell respiration is an example of something we call a metabolic pathway. The term metabolism really refers to all of the chemical reactions that take place within the cell. It's kind of the sum of all of the chemistry that happens within the cell. Metabolic reactions are usually organized into pathways. Here's an example of a very simple Metabolic pathway. We've got some starting molecule, we'll just designate A. And it undergoes a reaction where it's converted into something we'll call B. Okay? That particular reaction is regulated by a particular enzyme. And one of the things that we'll see is that enzymes are very specific, and this enzyme will only react with this reactant or substrate. Same thing. Reactant / substrate. B can then be converted into C. That requires a different enzyme. C can be converted into D. Okay? Well, that's a simple linear metabolic pathway. Here's a kind of a brief representation of some of the important metabolic pathways that occur within a cell. Looks a little bit complicated, doesn't it? Each one of these little dots right here represents an A, a B, a C, or a D. A substrate, a product, a substrate, a product. And one of the things that you'll see is that the metabolic pathways within a cell are pretty complicated, and they're all kind of intertwined with one another, and they are pretty much interdependent upon one another. This particular portion of metabolic pathways in the cell represents glycolysis. That's the partial breakdown of glucose. Anybody know what this little circular set of reactions represents? It's a part of cellular respiration. A set of reactions called the Krebs cycle. Okay? Well, if you look at all this kind of chaos, and one of the analogies I like to make sometimes is a pretty complicated street map almost. In some ways, metabolic pathways are kind of like streets. And the enzymes that regulate those are kind of analogous to traffic lights. And metabolism can be kind of made an analogy to the flow of traffic through streets in a big city. And things can go different ways depending upon whether traffic lights are red or green, for example. If you look at all this chaos, there are two basic types of metabolic pathways. One group of metabolic pathways are said to be catabolic. What are catabolic pathways? They tend to take things and break them down into simpler units. So complex to simple, basically. They tend to be energy-releasing reactions. Energy-releasing reactions can also be considered to be downhill, like that rock going down a hill. They are spontaneous. They happen on their own without an input of energy. And typically, during these reactions, molecules become oxidized. What's oxidation? You covered this in chemistry yet? We talked about oxidation? What happens to something when it becomes oxidized? It loses electrons. Lose electrons, oxidized. What other phenomena is always partnered with oxidation? Oxidation and reduction. Gain electrons, you become reduced. So typically what happens is these reactions occur in pairs. If something becomes oxidized, something else becomes reduced. This used to be one of our old chemists' favorite little acronyms, LEO-GR, a good way to remember oxidation and reduction. What's happening? Well, in biological systems, oxidation and reduction reactions oftentimes are associated with the gain or loss of hydrogen. What is a hydrogen? Look at hydrogen up here. Atomic number 1, right? What's that mean? It's got one what? One proton and one electron. So if you lose a hydrogen, are you losing an electron? Yeah. If you gain a hydrogen, are you gaining an electron? Yep. So in biological systems, oftentimes oxidation and reduction involves transfer of hydrogens. Okay? Oxidation reactions are associated with a loss of energy. Reduction reactions are associated with a gain of energy. Remember that I said that you should start thinking about hydrogens in terms of potential energy? So what's cellular metabolism really all about in the bottom line? It's about moving electrons and hydrogens around from one compound to another. Oxidizing one thing, reducing another thing. And that's how a cell accomplishes a lot of the energy transfer that's needed to do its work. The other major set of reactions are called anabolic. These reactions are synthetic in nature. What that means is it involves taking, for example, monomers and joining them together to make something more complex, a polymer. So starch synthesis would be anabolic. Starch hydrolysis would be catabolic. These reactions are energy requiring. They're uphill. They're like pushing that rock up the hill. Typically, they involve adding electrons or hydrogens to something, making it more reduced. They are not spontaneous. There is a strong relationship between anabolic and catabolic biochemical reactions in the cell. These oftentimes are coupled to these. These reactions provide the energy to drive these particular reactions. So that relationship is really very important. So where we're going to go next, and this is where it gets a little bit hairy, so stay with us on Friday and we'll make it through it. We said that catabolic reactions were spontaneous and anabolic reactions were not spontaneous. What does that mean? How does it what determines whether a reaction is spontaneous or not spontaneous? And that's going to get into a little bit of application of laws of thermodynamics and get you into this concept of free energy and delta G. So that's where we're going to be going tomorrow. Last minute words for lab? Any lab instructions, or are we good to go?
Just be on time.
Okay.
I'll be a little early.